Watch the back end of this sea cucumber. Nothing about it reads as dangerous. It's a soft brown tube, roughly the length of your forearm, easing across a rock ledge in the cold water off the Atlantic coast of Europe. No eyes worth mentioning, no teeth. It moves at about the speed of a slow leak. A crab has been picking at it for a couple of minutes now, the way you'd worry at a loose thread on a sleeve, and the cucumber has done nothing about it at all. Then the cucumber shifts, turns its rear end toward the crab, aims, and a spray of white threads comes out of its anus. Thin, distinct, separate threads. Dozens of them, fine as fishing line. And they don't drift the way you'd expect something soft to drift. They land. The instant they hit seawater, they begin to stretch. And the instant they touch the crab, they stop behaving like threads at all and start behaving like glue. The crab pulls one leg back. The thread comes with it. It grabs at the thread with a claw, which is exactly the wrong move because now the claw is stuck to the leg. 10 seconds, maybe 15. The crab is now a small knot of crab tied up in its own limbs, and the sea cucumber has already begun to move away slowly without ever having touched it. That's the footage. Here's the part that changes what you just saw. Those threads are not a weapon the sea cucumber grew. They're plumbing. They live inside the animal attached to the base of an organ called the respiratory tree, which is exactly what it sounds like. A branching, hollow structure that fills with seawater and pulls oxygen out of it. Sea cucumbers breathe through their backside, water in through the anus, into the tree, oxygen across the membrane, water out. It's their lung, more or less, and it's been doing that job for a very long time. The threads are moored to it, and when the animal is frightened, it ruptures the wall of its own cloaca and fires part of its breathing apparatus at whatever's bothering it. Think about what that costs. That's the shape of this whole thing, because when people talk about the ocean's most disturbing weapons, they tend to reach for the obvious hardware. The teeth, the jaws. The stingers built by evolution specifically to sting. And those are impressive, sure, but they're also honest. A stinger is a stinger. A tooth was always going to be a tooth. There's nothing unsettling about a machine doing the job it was made for. What's out here instead is stranger and, I'd argue, a lot more interesting. The creepiest weapons in the ocean are almost all conversions, secondhand equipment, organs that already had a full-time job, breathing, digesting, holding the body together, moving an eye, making skin slippery, making a pigment, and then got drafted into a second one, usually violently, usually under conditions where the animal had about a quarter of a second to decide. Every case in here follows the same 3 beats, whether the animal is a slug or a shark's nightmare. There's the original function, the thing the organ was built to do. There's the conversion, the moment evolution repurposed it. And then there's the bill, because a body doesn't get a free weapon. Something else in that animal has to go offline, or fall off, or grow back. Hold the sea cucumber for a second longer, because it pays that bill in public. The animal is a holothurian, one of about 1,700 described species, and it belongs to the same family tree as sea stars and sea urchins. Most of them do nothing more aggressive than eat sand. That's their whole career. They shuffle across the seafloor, pushing sediment into their mouths, digesting the organic film off it, and pushing clean sand out the other end. In some tropical lagoons, the resident sea cucumbers process an enormous fraction of the sediment every year. They are, functionally, the ocean's cleaning staff. Which is why what they do under threat is so hard to file. There are actually 2 versions of the trick, and it's worth keeping them apart because people mash them together constantly. The first is evisceration. Full evisceration. The animal contracts its body wall hard, tears its own internal membranes, and expels its guts. Its actual guts, the working digestive tract, with nothing decorative about it. Depending on the species, it either goes out the back, through the cloaca, or the animal ruptures near its own mouth, and the whole digestive tract goes out the front end. Some species can do it in under a minute. What comes out is a tangle of tubing, plus in many cases the gonads, plus one or both branches of that respiratory tree. So the animal is now missing its intestine, part of its reproductive tissue, and a chunk of its lung, all at once, floating in the water in front of a predator that now has a decision to make. And a lot of predators take the deal. That's the whole logic. A fish that's already committed to eating something suddenly has a warm pile of viscera in front of it and a fleeing sea cucumber behind it. And viscera are easier. The second version is the one in the footage, and it's more surgical. Certain species carry a bundle of specialized tubes anchored at the base of the respiratory tree. Holothuria forscali is the one that's been studied hardest, a dark, warty thing common in the eastern Atlantic and the Mediterranean. They're called Cuvierian tubules, after Georges Cuvier, the French naturalist who described them in the early 1800s and evidently found them worth naming after himself. At rest, they're short, coiled, sitting there doing nothing at all, which is a hint in itself because unused tissue is expensive tissue. When the animal is grabbed, muscles contract in the cloaca, the tubules snap free at their base, and water pressure inside the body cavity blows them out through a tear the animal makes on purpose in its own cloacal wall. The pressure does the work, and as they exit, seawater rushing past them, they elongate, reported at up to about 20 times their resting length. A tubule that started at roughly an inch can end up nearly 2 feet long, and it does that in a fraction of a second. Now, the stickiness. This is the part that got studied properly by Patrick Flameng and colleagues in Belgium, and the answer is not what most people assume. There's no glue in the ordinary sense, no secreted adhesive, no chemical setting reaction, no epoxy. The outer surface of the tubule is covered by an epidermis, and when the tubule hits a surface, that epidermis simply tears open on contact and the exposed inner material grips. It's mechanical. The adhesive strength comes from the material itself and from the enormous surface area of a 2-foot ribbon of tissue wrapping around a crab's leg, which is why it works instantly underwater on wet surfaces with no cure time, a combination that industrial chemists have been chasing for decades and mostly failing at. And once it's attached, it's very hard to remove. The tubule has almost no tensile give in one direction, and enormous give in the other. So pulling on it stretches it rather than freeing you. Struggling makes it worse. Anyone who has ever gotten a piece of packing tape stuck to their fingers understands the escalation curve here. Except the tape is alive, there are 40 of them, and you're breathing through your legs. So the sea cucumber walks away. Now the price. Those tubules were not spare parts. They were attached to the respiratory tree, and firing them means damaging the base of the organ the animal uses to breathe. The body wall has been torn open at the cloaca. Body fluid has been lost. And the tubules themselves are just gone, discarded on the seafloor, tangled around a very confused crustacean. Here's what the regrowth actually looks like, because this is where the accounting gets specific. Work published by Vandenspiegel, Jangu, and Flameng followed the regeneration of Cuvierian tubules in Holothuria forscali, and a single tubule takes about 5 weeks to come back completely. That breaks into roughly 2 days of repair right after the loss, then about 4 weeks of actual regeneration, then about another week of growth before its full size. But the interesting detail is the scheduling. When the animal has only expelled a few tubules, regeneration often doesn't start right away. There's a lag, and it starts later. When it has expelled a lot of them, the process kicks off immediately, and the tubules come back in waves, batches of roughly 10 to 30 at a time, with each new wave beginning about 10 days after the last. That's a body triaging. It doesn't rebuild all of it at once because it can't afford to. It rebuilds a partial magazine, then another, staggered, so that there's always something available while the rest is under construction. And through all of it, the cells doing the rebuilding aren't stem cells waiting in reserve. Existing cells in the 3 layers of the tubule dedifferentiate. They go backward, giving up their specialized identity, then divide, then re-specialize into what's needed. The animal is dismantling functioning tissue to rebuild a weapon it already spent. For the full evisceration, the bill is worse, and the mechanism is even harder to believe. An animal that has thrown its digestive tract at a predator no longer has a digestive tract. It cannot eat. Reduced appetite doesn't cover it. The equipment is lying on the seafloor behind it. It has to regrow an intestine from almost nothing. And it does. In Holothuria glaberrima, the Caribbean species that José García Araras and his group at the University of Puerto Rico have spent decades studying, a new gut grows out of the mesentery, the thin sheet of tissue that used to suspend the old one. A rudiment forms along that sheet and thickens. By around the 2nd week, a lumen appears, an actual internal channel, and the tissue layers organize themselves around it. Give it another week or so, and the proportions start to look like a normal intestine. A few weeks then, from nothing to a working gut, which is an absurd thing for an animal to be able to do, and it's also the only reason the defense makes any sense at all. You can only afford to throw away your organs if you're the kind of animal that can print new ones. That's the trade in its clearest form. What the sea cucumber really evolved was a return policy, and once it had one, it could afford to start spending organs. Keep that framework, because it's about to get applied to bodies that aren't nearly as forgiving, because the next one has a body that can't regrow much of anything, and it still throws away part of itself every time something with teeth gets close. Here's the clip. A baited camera rig, somewhere off the northern coast of New Zealand, White Island, or Great Barrier Island, or out at the Three Kings. Deep water, bad light, a frame full of scavengers working over a bait bag. And down in the middle of it, a hagfish, gray, eel-shaped, about the thickness of a garden hose, doing what hagfish do, which is nose into the food supply with an enthusiasm that borders on rude. A shark comes in and takes it. Full commitment. Mouth closed around the hagfish, and by any reasonable reading of the situation, that's the end of the hagfish. Then the shark's head disappears. That's what it looks like on the video. The whole front of the animal vanishes into a pale, expanding cloud. And a half-second later, the shark is backing off, mouth open, gill arches heaving in a way that anyone who has ever watched a dog eat a wasp will recognize instantly. It's gagging. It's trying to clear something out of its throat that it has no equipment for clearing. And the hagfish, entirely uninjured, swims out of the cloud and goes back to the bait. Vincent Zinsen and his colleagues at the Museum of New Zealand Te Papa Tongarewa published that footage in 2011. 14 separate recorded attacks by sharks, dogfishes, conger eels, cusk eels, scorpion fishes, wreck fishes, different predators, different depths, same outcome almost every time. The hagfish is bitten, the hagfish slimes, the predator lets go. And what it's throwing at them started life as skin. That's worth slowing down on, because it's easy to hear slime and picture something like spit or snot, a secretion, a fluid, a thing an animal makes and squirts. That's not the category this belongs to. Every fish in the ocean has mucus glands in its skin. That's standard equipment. It's what makes a fish slippery in your hands, and the slipperiness is only the surface of it. The coating manages water and salt across the body surface, carries antimicrobial compounds, and makes the animal harder for parasites to settle on. It's a boundary layer. It's the reason skin works. The hagfish took that boundary layer and industrialized it. Run your eye down the side of one and you'll see a row of pores, evenly spaced, from just behind the head all the way to the tail. There's a matching row on the other side. Depending on the species, you're looking at somewhere in the neighborhood of 100 of them or more, and each one is the opening of a slime gland, a deep, purpose-shaped sac packed with two kinds of cell that do nothing but load ammunition. And in 2023, a team working on hagfish epidermis published something that closes the loop on where this came from. They looked at ordinary hagfish skin, regular epidermal cells, not gland cells, the boring stuff between the pores, and found that those cells contain miniature versions of the same threads found in the slime, small ones, coiled, in the same fashion, doing something much humbler. The specialized glands appear to be an enlargement of a thing hagfish skin was already quietly doing everywhere. That's the conversion, in as clean a form as you'll find it anywhere in this. A general purpose feature of skin, present across the body, got concentrated into 100 dedicated bunkers and scaled up until it could disable a shark. Now the mechanism, because the physics of this is legitimately strange. Inside each gland, there are two cell types. The first is called a gland mucus cell, and it's full of vesicles packed with mucin, the same class of molecule in the mucus you already have in your own airways and gut. The second is called a gland thread cell, and it's the one that makes this whole thing work. Each gland thread cell contains one thread, a single continuous protein filament, wound up inside the cell in tight, ordered loops, like thread on a spool, except the winding is far more precise than that. Unwound, one of these threads runs somewhere in the range of 2 to 12 inches long, and it's on the order of 1,000th of a millimeter thick. Call it 1/50th the width of a human hair, and about 1 foot long, coiled inside a single cell you'd need a microscope to find. The coiled bundle is called a skein. And when the animal fires, the gland squeezes and the cells are ejected whole, a mode of secretion called holocrane, which is a polite biological term for the fact that the cell itself is the product. The whole cell gets expelled and destroyed in the act of delivery. Every gland thread cell a hagfish fires is a cell it will never get back. What leaves the pore is a thick, concentrated, unremarkable-looking exudate. Barely anything, a dab. Then the seawater does the rest. The mucin vesicles hit salt water and burst, and the mucin swells enormously. These are molecules built to hold water, and they hold an obscene amount of it. At the same time, the skeins begin to unravel, and the unraveling is not passive. Work by Wingard and Fudge, and later by Bernards and colleagues, sorted out the two pieces of it. The skein is held together by a protein adhesive that dissolves in seawater, so contact with the ocean releases the coil. And the mucin strands, swelling and stretching in the moving water, transmit force to the loose end of the thread and pull it out. The mucus is what deploys the fiber. The two components only work as a weapon because they're released together and mixed by the flow that the predator itself is generating. Result: a dab of material becomes a three-dimensional net of fine protein threads spread through a volume of water, and reported expansion figures run to something on the order of 10,000 times the original volume. A single pinch on the tail of an adult Pacific hagfish has been measured producing close to a quart of dilute slime. That's roughly 7 times the volume of the entire animal, out of a few glands, in well under a second, and the slime is almost entirely water. That's the trick and the elegance of it. The solid fraction is minuscule. The threads and the mucin together are a rounding error by weight. What's been built is a scaffold, an open network fine enough that water can't easily move through it. It doesn't have to be strong, it has to be everywhere at once and hard to pump, which is exactly the property that makes it lethal to a gill. A fish breathes by moving water in one direction across the gill filaments, continuously, in through the mouth, across the arches, out the sides. The whole system is built around low resistance and steady flow, and it's an unforgiving design because a fish's gills are also structurally delicate, huge surface area, extremely thin membranes, packed close together. Anything fibrous that gets in there fouls the flow. Zinssen's group put it plainly, the slime affects gill-breathing predators by clogging the gills, most likely by increasing resistance to water flow. So the shark bites, and in doing so, it inhales. It pulls water into its own mouth, and in that instant, the hagfish converts the water inside the shark's head into a net. The predator's breathing apparatus stops working, while its mouth is still closed on the meal. Every single one of those recorded predators released the hagfish. Then they sat there convulsing their gill arches, trying to cough with anatomy that has no capacity for coughing. There's a second-order problem here that the hagfish had to solve, and it's the part that convinces me this is a genuine weapon rather than a lucky accident. The hagfish is inside the cloud too. It has gills. It just filled the surrounding water with the exact material that shuts gills down at point-blank range, with itself at the center of the blast. So it has two moves for that, and both of them are documented and both are ridiculous. The first, it ties itself in a knot, an overhand knot, near the tail, and then it runs that knot forward along its own body like a squeegee, pushing the slime off the skin and giving itself something to brace against while it pulls free of whatever's holding it. The knot is a real behavior with real uses. Hagfish use the same trick to tear food off a carcass, bracing against their own body because they have no jaws to lever with. The second, it sneezes. It has a single nostril, and it forces a sharp pulse of water and slime out through it to clear the passage. A jawless fish, several 100 million years of lineage behind it, blowing its nose in an emergency. Now, the bill. Start with the cells, because holocrine secretion is brutal accounting. The gland doesn't leak a product and refill a reservoir. It destroys its own cells to deliver them. Every deployment strips the gland of a population of mature thread cells and mucus cells that have to be rebuilt from immature cells at the base of the gland, grown, filled, and in the case of thread cells, wound. That last part is the expensive one. A gland thread cell has to synthesize a foot of intermediate filament protein and then coil it, in order, without tangling it, inside itself. That is not a fast process. Published estimates for a hagfish to fully recharge its slime supply run in the range of several weeks. Run that forward. A hagfish that has just saved its own life from a shark is now, for the better part of a month, a soft gray tube living on the seafloor with a partial magazine. It is not less hungry during those weeks. It still has to go into carcasses. It still has to compete with the same predators that just tried to eat it, at the same bait falls, in the same dark. There's a protein cost on top of that. Those threads are made of intermediate filament proteins, keratin relatives, and nitrogen is not cheap for a deep-sea scavenger with an unpredictable food supply. The hagfish is converting hard-won dietary protein into fiber it intends to throw away. If you want a sense of what that adds up to at scale, there was an accident in Oregon in the summer of 2017 that made the point better than any laboratory could. A truck hauling live hagfish overturned on Highway 101 near Depot Bay. The animals were bound for export, since they're eaten in South Korea, and their skin is what most eel skin leather actually is. Roughly 7,500 pounds of stressed hagfish came off the truck onto a public road. They did the only thing they know how to do. The highway had to be closed and washed down with a fire hose, and the photographs are, frankly, difficult to interpret at first glance, because the road appears to be covered in about a foot of gray pudding. That's a slime supply from one truckload of frightened fish, on land, with no help from ocean currents. And every animal in that pile spent weeks of protein and thousands of destroyed cells to produce it, in response to a threat that wasn't even a predator. Skin's original assignment is to keep the outside on the outside. The hagfish took that job description and inverted it. It now uses the boundary layer as an offensive weapon that fills the space around it, and it pays for the privilege in cells that die on delivery and a month of running on empty. Both of those animals are spending organs. Let's go one level deeper into the body, to the thing that holds organs in place at all. Night dive, sand flat, a light sweeping across the bottom. A brittle star is out in the open, which is a mistake, and it's making the mistake because it's feeding. 5 long arms, thin as wire, and a small central disc about the size of a nickel. The arms are doing this constant serpentine flicking that makes the whole animal look like it's being electrocuted at low voltage. A fish comes in from the side and takes an arm. Clean bite halfway down. And what happens next is over so fast that on video, you have to step through it frame by frame. There's no tug of war. There's no stretching. The arm separates at a joint several segments closer to the disc than where the fish actually bit, well inside the animal in undamaged tissue, and the brittle star is already moving. 4 arms rowing, gone under a rock in under 2 seconds. The severed piece is still writhing. That's the part that stays with you. It's lying on the sand, coiling and flexing with real coordination for a good while after it stopped being part of anything. The fish stays on it. Of course it does. There's a moving object in front of it. Now everybody knows lizards drop their tails. Most people file this under the same heading and move on. It isn't the same heading, and the difference is the entire reason this belongs in here. A lizard's tail has a fracture plane, a built-in weak point in the vertebrae, a designed break. That's a structural feature. What echinoderms have is something else. And once you understand it, you start seeing it everywhere in the group. They have connective tissue they can command. Think about what connective tissue does in your body. Collagen, ligaments, tendons, the sheets and cables that hold your skeleton together and anchor muscle to bone. The entire value of that material is that it's dumb and it's reliable. It has one mechanical state. Your knee ligaments do not have a setting. They're rope, and rope's virtue is that rope stays rope. Echinoderms broke that rule about 500 million years ago, and they've been exploiting it ever since. Their connective tissue is called mutable collagenous tissue. You'll also see it called catch connective tissue, and it can change its stiffness by an enormous factor in seconds under direct nervous control. Here's roughly how it works. You've got collagen fibrils sitting in a gel-like matrix that's rich in proteoglycans, and the fibrils are linked to one another by cross bridges of other proteins. The stiffness of the whole tissue depends almost entirely on how firmly those fibrils are stuck to each other, because that's what determines whether they slide when you pull. And sitting throughout that tissue are cells called juxtaligamental cells. They're stuffed with secretory granules. They're wired directly to motor neurons, and their job is to dump chemical factors into the matrix that either strengthen those cross bridges or dissolve them. Researchers have pulled out proteins that do each side of it, stiffening factors and softening factors, with names like tensilin and softenin, which are among the more honest names in biology. So the tissue has settings: soft, standard, stiff, and a nerve impulse can move it between them. Before we get anywhere near the weapon, look at what that buys an animal in ordinary life, because this is the original job and it's a good one. A sea urchin can lock its spines, mechanically lock them at the ball joint by stiffening the collagen sleeve that wraps it. Try to push the spine sideways and it doesn't yield. And the urchin is spending nothing to maintain that. No muscle contraction, no metabolic cost, no fatigue. It's a mechanical latch made of ligament, and it can hold indefinitely. Same principle keeps a sea star clamped to a rock through hours of surf. Same principle lets a brittle star wedge itself in a crevice so firmly that you'll tear it before you extract it. A muscle holding that posture would burn energy the whole time and eventually give out. Stiffened collagen holds for free. That's the real function. Postural economy. An entire phylum that figured out how to hold a position without paying for it. The weapon is that exact same control system run all the way to the other end of the dial, at a specific location, on purpose. There is a defined autotomy plane, a real anatomical structure. At that plane, the tissue holding the arm to the body is mutable collagenous tissue with an unusually dense population of juxtaligamental cells. In brittle stars, the specific parts that let go are the tendons connecting the intervertebral muscles to the vertebral ossicle, and those tendons are mutable collagenous structures sitting in direct contact with juxtaligamental cell processes. The starfish Asterias rubens has been worked out in similar detail and is generally treated as the paradigm case for how the plane is organized. The animal sends a signal. The cells release their factors. The cohesion between collagen fibrils at that plane collapses, and the tissue that was holding the arm on simply stops being able to hold anything on. The arm gets released, under orders, by a tissue that chose to stop working. Nothing tore, nothing frayed, and if you look at the stump under magnification, you find a clean, organized surface at a predictable anatomical location, with the body wall already closing over it. Which raises the obvious question of why the severed arm keeps moving. And the answer is that echinoderms don't centralize the way we do. There's no brain. There's a nerve ring around the mouth and a radial nerve cord running the length of each arm, and that cord is perfectly capable of generating coordinated movement on its own. Separate the arm and the nerve cord in it keeps firing. It doesn't know. It goes on producing the same locomotor rhythm it was producing a minute ago, in the sand, in front of a fish, which is precisely the outcome the whole system is designed to buy. And this is not a rare emergency measure. That's the thing that surprised me most when I went looking at the survey data. In brittle star populations, arm loss is closer to a baseline condition than an event. Studies that pull up samples and score them routinely find large fractions of the animals carrying at least one arm in some stage of regrowth. In plenty of surveys, most of them. Sublethal predation is constant. Fish and crabs crop brittle star arms the way you'd graze a lawn, because the arms stick up out of the sediment while the disc stays hidden. And the arms are the only part on offer, so there's a whole population of animals down there in a permanent state of partial rebuild. So here comes the bill, and the sea stars make the bill more legible than the brittle stars do, because of how a sea star is organized. A sea star arm is not an appendage in the way your arm is an appendage. Open one up, and you find that the arm contains a pair of gonads and a pair of pyloric caeca, the digestive glands, the organs that actually process food and store the energy reserves. In a 5-armed sea star, something like a fifth of the animal's reproductive tissue and a fifth of its digestive and storage capacity lives inside each arm. Drop an arm and you've dropped a fifth of your gut and a fifth of your capacity to reproduce this season, along with the muscle, the skeleton, the nerve cord, the tube feet, and the water vascular plumbing that ran through it. Then you have to rebuild all of it, with less digestive tissue than you had yesterday. That's the ugly part of the arithmetic, and it applies across the group. The animal is now in a rebuilding project that requires energy, while carrying reduced equipment for acquiring energy. For a brittle star, the arms are also the feeding apparatus, since many species raise them into the current and catch particles on them, and they're the means of locomotion. Fewer arms means less food caught and slower movement, which means slower regrowth, which extends the window in which it has fewer arms. Regeneration timelines depend heavily on species, temperature, and food supply, and the honest range is wide. For many brittle stars, you're looking at months to get a functional arm back to size. For sea stars, it's often longer. And at the far end of that scale, there's the phenomenon that gave the whole thing its nickname: Some sea stars in the genus Lynchia can go past arm loss into something much more extreme. A single detached arm with a small fragment of the central disc, or in some cases without one, can regenerate an entire animal. 4 new arms bud from the base of the old one, tiny at first, and for a long stretch of that process the thing looks like a bright object with a tail. Divers call them comets. The whole rebuild can run to a year or more. A year. To reconstitute a body out of a limb. While we're inside a sea star, there's a second conversion in the same animal that's worth 30 seconds because it's the same principle applied to a completely different organ. Many sea stars feed by turning their stomach inside out. The cardiac stomach, an internal digestive organ, the same category of thing you're carrying around right now, gets pushed out through the mouth, spread over the prey, and used to digest food outside the body entirely. With a clam, this gets uncomfortable to think about. The sea star humps over the shell, gets its tube feet on both valves, and pulls. It doesn't need to win. It doesn't need to open the clam. It needs a gap. And the reported gaps are on the order of a fraction of a millimeter, a hair's width of daylight between the shells. Then it slides its stomach through that gap and starts digesting the clam inside its own shell. The clam's defense is a fortress with one door, and the sea star's answer is to pour part of its digestive system under the door. Same theme as everything else here. An organ evolved to hold food and break it down, used instead as an offensive instrument outside the body, in the water, in contact with a live animal that's actively trying to stay shut. The connective tissue story is the cleaner example, though, and it says something larger about how these animals are built. The material holding the body together is under the same kind of control as a muscle. Structure became a decision. And once your structure is a decision, you can decide to let part of yourself go, which is a spectacular defense and a permanent liability at the same time, because a tissue that can be told to fail is a tissue that must never receive that signal by accident. So far, every animal in here has been spending its own hardware. Now watch one spend somebody else's. Shallow reef, good light, middle of the day. There's a slug on an orange sponge, and it is making no attempt whatsoever to be subtle. Bright white body, a rim of gold around the edge, 3 black stripes running down its back. It's maybe 1.5 inches long. It has no shell. It has no speed. It's a snail. It moves at snail rates, and it's crawling across an open surface in full daylight on a reef that contains wrasses, triggerfish, pufferfish, and about 40 other things that eat invertebrates for a living. A wrassie swims past it, looks at it, keeps going. That happens over and over on nudibranch footage once you start watching for it, and it's the anomaly that has to be explained. Because this animal has given up every conventional defense a mollusk has. Its ancestors had a shell. Somewhere back down the line, sea slugs abandoned it. The whole calcium fortress, the thing that made the group successful in the first place, and what's left is a soft, slow, brightly colored, entirely edible-looking package with no cover and nowhere to hide. Animals that do that usually have a reason. The reason here is that the slug is carrying a payload, and it did not manufacture that payload. It ate it. The sponge underneath it is the supplier. Sponges have a problem you don't often think about, which is that they can't move, can't hide, can't heal quickly, and are essentially a bag of nutritious tissue bolted to a rock in a crowded neighborhood. Their solution, developed over an extremely long time, is chemistry. Sponges are the most prolific chemical factories in the ocean. A meaningful share of every marine natural product that pharmaceutical companies have ever screened came out of a sponge. And the reason those molecules exist is not that sponges are being generous with drug discovery. They exist to make the sponge inedible, to stop other organisms from settling on it, and to kill things that try to grow into it. So a sponge is a block of poison. and the poison works, mostly, which means any animal that can solve the chemistry gets a food source with almost no competition. That's the deal the Dorid nudibranchs took. Take the compound that's been worked out best, latrunculin A. It comes from sponges. It was first pulled out of the Red Sea sponge Latrunculia, which is where the name comes from, and it turns up in others, including Spongia mycophigiensis in Fiji. Chemically, it's a macrolide, a big ring-shaped molecule, and what it does inside a cell is specific and severe. It binds to actin. Actin is one of the most fundamental proteins in complex life. It's the cytoskeleton, the protein that assembles into filaments and gives cells their shape, lets them crawl, lets them divide, lets muscle contract, lets your gut cells hold their structure. Actin has to be able to polymerize and depolymerize constantly. That dynamic assembly is the machinery of being a cell. Latrunculin grabs the individual actin units and prevents them from joining the chain. Assembly stops. Cells lose shape, lose the ability to move, and in short order, lose the ability to function. You want the measure of how good this molecule is at that job? Cell biologists keep it on the shelf. Latrunculin A is a standard laboratory reagent. If you're a researcher who needs to switch off actin polymerization in a dish so you can see what a cell does without a cytoskeleton, this is the compound you order. A sponge in the Red Sea worked out a precision inhibitor of one of the most conserved proteins in the animal kingdom, and human labs now buy it in vials. The slug takes that molecule out of its meal, alive, unbroken, and stores it. Every part of that sentence is a problem to be solved, and the solutions are what make this interesting. Digestion is a demolition process. The whole purpose of a gut is to take complicated molecules and reduce them to simple ones. A gut that transports a complex ring compound across its wall without dismantling it, and then hands it to a courier system for delivery to a specific piece of skin, is doing something a gut was never designed to do. That's the conversion here. The digestive system stopped being purely a demolition site and became a sorting facility with an inventory. And the sorting is selective, which is the part that turns a curiosity into a strategy. There's a study called Choose Your Weaponry, a title I have some affection for, where researchers looked at closely related Chromodoros species and found that out of the whole cocktail of compounds available in the sponges they were eating, the slugs were preferentially accumulating latrunculin A specifically, related species, overlapping diets, and the same molecule selected out of the mix. That's procurement. The animal is choosing its ammunition out of a mixed shipment. Then there's where it goes. And this is the detail that made me sit up. Researchers ran mass spectrometry imaging on Chromodoros. It's a technique that lets you map where a specific molecule sits inside a tissue, essentially a chemical photograph. Latrunculin A showed up through the mantle tissue and in the mucus glands, and it was heavily concentrated in the vacuoles of structures called mantle dermal formations. Those are specialized storage glands, globular, packed into the mantle skin, full of cells with big, empty-looking vacuoles that are in fact full of stolen sponge chemistry. And the distribution across the animal isn't even. The concentration is highest in the rim of the mantle, the edge. The outermost margin of the animal, the part that flares out around the body, the part that a fish's mouth reaches first. The slug has mapped its poison onto its own outline. That's a defended perimeter, built from a molecule it stole, positioned according to the geometry of how it's likely to be attacked. And it fits how these attacks actually go. A fish doesn't usually commit to swallowing an unfamiliar invertebrate in one motion. It mouths it, tastes it, and decides. Which means the defense doesn't need to be lethal. It needs to be concentrated at the point of first contact, and it needs to be foul enough to trigger rejection inside a second. Which leaves one very awkward problem. If this compound shuts down actin in animal cells and the slug is made of animal cells, Why isn't the slug dead? Nobody has fully closed that one, and I'd rather tell you it's open than pretend otherwise. The leading idea, and there's biochemical and molecular work testing it, is target site resistance, that the slug's own actin is modified in some way that stops latrunculin from binding it properly. Same principle as an insecticide-resistant mosquito with an altered nerve channel. That's a hypothesis with evidence behind it, currently being worked on. It hasn't been nailed shut. There may also be sequestration doing part of the work. If the compound stays packaged inside vacuoles and dedicated storage cells and never floats free in the tissue, it can't reach the actin it would otherwise attack. Probably both. That's usually how these things resolve. Now the colors? Since we're here, and everybody asks. The standard explanation for a bright, patterned, slow-moving animal is aposematism, warning coloration. The same logic as a wasp or a poison frog. Honest advertising that says the cost of eating me exceeds the benefit. For nudibranchs, the evidence is real but patchier than the popular version implies. Some species clearly work that way, with reef fish showing learned avoidance of specific patterns. Others are gaudy in ways that are hard to read as signals to fish at all, and some are effectively camouflaged against the exact sponge they eat, which is the opposite strategy in the same body plan. It's a mixed picture, and anyone telling you every colorful nudibranch is a billboard is tidying up the data. Now, what does this cost? It costs the slug its entire freedom of choice about food, and that turns out to be enormous. Dorid nudibranchs of this kind are among the most extreme dietary specialists in the ocean. Many feed on a single genus of sponge. Some are down to one species. Their entire defensive apparatus is downstream of one prey item, which means the animal cannot switch. Its weapon and its dinner are the same object, and its resistance mechanisms, its storage tissue, its sorting machinery are all tuned to a chemistry that only one organism on that reef produces. Now take that sponge away. Bleaching event, disease outbreak, a dredge, temperature anomaly, a change in water chemistry. Pick your century, they're all happening. The slug loses its food and its defense in the same stroke, and it has neither a shell to fall back on, nor the speed to run, nor a generalist gut that could handle anything else. There's no fallback position because the fallback position was traded away a long time ago in exchange for a poison that made the fallback unnecessary. And here's the layer of this that I find hardest to shake. That molecule was itself already a weapon. The sponge didn't build latrunculin for the slug's benefit. The sponge built it to stop things from eating it and settling on it. It's a defense that failed at its one job in one specific case against one specialist that cracked it. And the specialist then picked it up off the floor and wore it. So the chain runs. A molecule invented to protect a sponge, defeated by a slug, then carried by that slug into its own skin and pointed at fish. Third hand. The compound is doing the same work it always did, guarding a soft body from being eaten. It just changed employers. That pattern is about to get pushed a great deal further by the slug's own relatives. They stopped stealing chemicals and started stealing loaded weapons. The relatives are called aelids, and you can tell one on sight. Where the sponge eaters are built like padded ovals, an aelid looks like a slug wearing a coat of flames. Its whole back is covered in soft fingers. Chirata is the anatomical term. Dozens of them, sometimes more than 100, in oranges and violets and impossible blues, swaying in the surge like hair. Those fingers are the arsenal, and what sits loaded in their tips was manufactured by another animal for that animal's own defense and then taken from it in working condition. To see the theft happen, watch one eat. The prey is a hydroid, a colonial cousin of jellyfish and sea anemones that grows across rocks and pilings in branching tufts, so that most divers swim past it thinking it's a plant. It is not a plant. Each of those fronds carries rows of polyps, and each polyp is studded with one of the most sophisticated pieces of cellular machinery that has ever evolved: the stinging cell. It's worth being precise about what a stinging cell actually is, because the popular image, a bit of skin that's irritating to touch, does not begin to cover it. Inside the cell sits a capsule called a nematocyst, and inside the capsule sits a hollow thread, tipped and lined with barbs, folded inside out and coiled under pressure. The osmotic pressure inside that capsule has been measured at something on the order of 150 atmospheres. For scale, that's several times the pressure inside a champagne bottle, held in a container smaller than a red blood cell. When the trigger fires, a touch on a hair-like sensor, a chemical cue, usually both, the capsule pops open and the thread everts, turns itself right side out at the target, like the finger of a rubber glove being blown inside out, except armed. High-speed work on Hydra, the little freshwater cousin of these animals, clocked the discharge at around 700 billionths of a second, with the barbed tip experiencing accelerations in the millions of G. As far as anyone has measured, it is one of the fastest movements in the animal kingdom. Maybe the fastest. The tip hits with pressures in the range of what a bullet delivers, which is how something softer than gelatin manages to punch through the armored shell of a crustacean. And it's a single-use device. That's the design constraint that shapes everything else in this story. A nematocyst cannot be rearmed, rewound, or repacked. It fires once, it's spent, and the animal that built it throws the cell away and grows a new one. A jellyfish sting isn't an attack in any muscular sense. It's a few thousand of these going off at once, a volley from an animal that is, in every other respect, barely more organized than wet tissue paper. So a hydroid colony is a hedge of loaded, hair-triggered, disposable guns. The aeolid crawls onto it and starts eating, slowly, deliberately, mouth on polyp, rasping away with its radula, chewing through a structure that is defended, at essentially molecular resolution, against exactly this. And here's the first honest gap in the story. Nobody can fully explain how the slug does that without being shredded from the inside. Aeolids secrete heavy mucus while they feed, and the leading idea is that the mucus either physically sheathes the stingers or chemically talks them out of firing. Some capsules probably do go off during the meal, and the slug seems able to absorb that cost. The details are still being worked on, and anyone who tells you it's fully solved is ahead of the data. What's certain is the outcome. The polyp goes in. Most of its ammunition doesn't fire. Now follow that ammunition through the slug, because this is where the anatomy turns strange. In an aeolid, the digestive gland, the organ that in you would be something like the liver, is not a single lump. It branches. Ducts run from the gut out into every single one of those fingers on the animal's back, all the way to the tips. The slug's digestive system reaches into its decorations. Food doesn't just get processed in this animal, it gets routed. Soft tissue from the hydroid is digested along the way, like any meal. The intact nematocysts are not. They ride the ducts outward, through the branches, into the serrata, and at the very tip of each one, there's a chamber called a cnidosac. Waiting inside it are specialized cells, cnidophages, literally stinger eaters, that swallow the stolen capsules whole and then don't digest them. They hold them, aimed outward. Stop on that for a second, because it's easy to let it slide past. A cell from one animal, a loaded single-use weapon still primed to fire, is sitting alive adjacent and functional inside the body of a second animal in a purpose-built holster pointed at the outside world. Biologists call the stored capsules kleptocnidae, stolen stingers. The name is not a metaphor, and the stocking of those holsters is not random. In the species that have been studied closely, the slugs are selective about which capsule types they keep. Hydroids carry a mixed inventory of stinger classes, some built for punching, some for tangling, some for gluing. and the slugs preferentially warehouse certain kinds over others. There's evidence in some species that the big penetrating types are favored, the ones that actually hurt. Whether the slugs can take in immature capsules and finish maturing them in storage is an open question that researchers are still arguing about. The selectivity itself is not. This is procurement again. The same behavior as the sponge-eating cousins choosing their molecule, except now the item being selected off the shelf is a machine. Then a fish makes a mistake. It bites a cerus, or mouths one, the standard taste test. The cnidosac at the tip ruptures under the pressure, and the stored nematocysts discharge into the mouth that's biting down on them. The threads fire, the barbs land. The toxins that a hydroid brewed to defend a colony on a rock go to work inside the palate of a fish that has never touched that hydroid in its life. The sting arrives secondhand, re-aimed at the exact geometry of the attack, in the single most sensitive tissue the attacker owns. Fish spit them out. That's the record over and over in feeding trials and in the field. The bite, the recoil, the slug rejected with damage that a soft-bodied animal can usually survive. And some aeolids stack a second trick on top, one you've already seen in this story wearing a different body. The serrata can be dropped. Grabbed hard enough, some species simply release the fingers under attack. The same commanded self-amputation the brittle stars run, and the shed serrata writhe on their own, and they can still sting because the cnidosacs go with them. The attacker is left mouthing a twitching armed decoy while the slug pours itself into a crevice. Now the bill, because this weapon has a property none of the others so far have had. It expires. A nematocyst is a spring under load inside a dying cell. The slug didn't build it, doesn't own the blueprints, and cannot service it. From the moment a capsule is stolen, it's on a countdown. Stored kleptocnidae stay functional for a while, on the order of days to weeks, depending on the species and the conditions, and then they degrade and have to be replaced. Wholesale. The entire arsenal, on rotation, forever. Which means the slug is chained to the treadmill. It has to keep eating hydroids, has to keep grazing the minefield, not just for calories, but to keep its guns loaded, because an aeolid that goes too long without dangerous food becomes what it appears to be, a soft, slow, brightly colored snack with no shell. The weapon and the meal are the same object again, the same trap the sponge eaters walked into, except with a fuse attached. Diet lock-in with a countdown timer. And at the far end of this strategy, out in the open ocean, there's a species that pushed it to a place that reads like fiction. It's called Glaucus atlanticus. An inch or so of nudibranch, silver on one side, deep electric blue on the other, with serrata fanned out in clusters like splayed hands. People who find them call them blue dragons, and the photographs look retouched, even when they aren't. It lives at the surface of the open sea, upside down, hanging from the underside of the water's skin, kept afloat by a bubble of swallowed air it holds in its stomach. The blue side faces up, camouflage against the water for birds looking down. The silver side faces down, camouflage against the bright sky for fish looking up. It cannot really swim. It drifts where the wind pushes the surface, full stop. It drifts there because that's where its prey drifts. Glaucus eats the Portuguese man-o'-war. The man-o'-war is the most notorious stinger at the ocean's surface, a colonial animal flying a gas-filled sail, trailing tentacles that can run tens of feet, armed with nematocysts potent enough to put a swimmer in the hospital, and rarely worse. Fish avoid the tentacles. People avoid the beach when the sails blow in. And this slug, an inch long, with no shell and no speed, rides the same winds, finds the same animal, hauls itself along the tentacles, and eats them. Then it does the aeolid thing with the inventory, and it does it with the man-o'-war's heaviest ordnance. The most potent capsule types get routed out to the tips of those splayed hand serrata and concentrated there, packed denser than they ever sat in the tentacle they came from. Which produces the detail that lifeguards on Australian beaches have learned the hard way, because these animals strand in numbers when the wind is wrong. The sting of the blue dragon is reported to be worse than the sting of the man-o-war it robbed. The slug is not just carrying stolen weapons. It's carrying a curated concentration of the best ones, in a package small enough that people pick it up because it's pretty. The price is the whole shape of its life. Glaucus gave up steering. It's a passenger of the wind, permanently, an animal whose entire existence is parked downwind of its food, and when the weather turns, whole armadas of them wash up and die on sand they never chose. That's the trade at its most naked. The best weapons at the surface of the sea, in exchange for never again deciding where you're going. The treadmill, though, the expiring ammunition, the forced diet, the arsenal that has to be perpetually restolen. That problem has a second solution, and a crab found it. Don't take the ammunition out of the gunner. Take the gunner. Alive. There's a crab in the Indo-Pacific, in the Red Sea, and across the reefs east of it. That's small enough to sit on your thumbnail. Boxer crab is the common name. Pom-pom crab is the other one, and both names come from the same observation. The crab appears to be holding something in each claw, and it will not put them down. Look closer, and the things it's holding are alive. They're sea anemones, tiny ones. One gripped in each claw like a pair of fluffy gloves, and when anything threatening approaches, a fish, another crab, a diver's forceps, the crab raises them and jabs, leads with them, fights with them, presses living, stinging animals into the face of whatever's bothering it, like a riot cop with a taser in each hand, except the tasers are also independent organisms with their own agenda. An anemone is a relative of everything the aeolids have been robbing. Its tentacles are packed with the same nematocysts, the same single-use spring-loaded harpoons, the same toxins. The slug's approach was to disassemble that animal and warehouse the parts. The crab's approach is to skip the disassembly, keep the whole weapon system intact and running, feeding itself, maintaining itself, regrowing its own ammunition on its own schedule, and just hold on to it permanently, because this is not a behavior the crab does sometimes. Crabs of the genus Libya are essentially never found empty-handed. Survey after survey, wild-caught animal after wild-caught animal, the claws are occupied. The association is so complete that the crab's own body has been reshaped around it. Those front claws are slender, delicate, specialized pincers, tweezers built for gripping soft anemone tissue without tearing it, and they've become close to useless for anything else. The crab cannot really feed itself with its own claws anymore. It eats with its other limbs, picking and passing food to its mouth with the walking legs because the hands are full. The hands are always full. An arms race pun would be cheap here. So instead, look at what the arrangement costs each side, because this is not a friendly partnership, and the closer researchers have looked, the worse it gets for the anemone. A team at Bar Ilan University in Israel, Yisrael Schnitzer, Yair Achituv, and colleagues spent years on Red Sea boxer crabs, and their findings turned a cute reef anecdote into something considerably darker. First, the crabs control what their anemones get to eat. An anemone in the wild catches food with its tentacles. An anemone in a crab's claw has its catch stolen out of its tentacles by the crab, routinely. The crabs keep them, in effect, on rations. And the anemones held by crabs are dwarfed, measurably smaller than the same tissue would grow free living. Kept small the way a bonsai is kept small because a small anemone is a manageable weapon and a large one is a handful in the wrong sense. The researchers could take a crab's anemone away, let it live free and watch it grow. In the claw, it doesn't get to. Second, and this is the finding that got the study attention far beyond crab biology. What happens when a boxer crab loses one of its anemones? A crab with one glove is a crab with a problem. So it does something that, as far as anyone knows, almost nothing else in the animal kingdom does. It takes its remaining anemone and tears it in half, deliberately. Over the course of minutes, working the tissue apart with those tweezer claws, splitting one living animal into two ragged pieces, and then it holds one piece in each claw and waits. Anemones are cnidarians, and cnidarians regenerate. Within days, each half has healed into a complete, functional, smaller anemone. The crab has cloned its own weapon, induced asexual reproduction on demand in a different species as an act of inventory management. And if a crab has no anemone at all, it finds another crab that has them, and it attacks it. The point of the attack is robbery. Fights between boxer crabs over anemones are the documented norm, and they usually end with the loser giving up a fragment, which both parties then split and regrow. Which leads to the strangest census result in this whole story. When the Israeli team genotyped the anemones being carried on the reef, crabs' left and right anemones were overwhelmingly clones of each other, and they struggled to find this kind of anemone living free on the reef at all. It's possible, and the researchers raise this carefully, that in that system, the anemone barely has an independent existence anymore, that its population substantially lives in custody, propagated by tearing, passed from claw to claw down generations of crabs, like a weapon that exists mostly in armories. Nobody knows what the anemone gets out of this, if anything. Transport, maybe? Scraps, maybe? The honest answer is that from the anemone side of the ledger, this looks less like symbiosis and more like livestock. An animal kept alive, kept small, kept hungry, bred by mutilation, and spent as a weapon whenever something larger comes close. The crab's bill is subtler but real. It has mortgaged its own hands. A predator that specializes this hard on a held weapon has forfeited the general purpose clause that every other crab its size uses to feed, dig, fight, and climb, and it has tied its security to an object that can be dropped, stolen, or die. A disarmed boxer crab isn't just weaker. It's an animal whose anatomy assumes a tool it no longer has, which is why the robbery behavior exists. The alternative to stealing a replacement is being a crab with tweezers in a knife fight. Hold the shape of what just happened, because the escalation across these last three animals is worth seeing plainly. The sponge-eating slugs stole a molecule. The iolids stole a loaded machine. The boxer crabs stole the factory, the entire living manufacturer, maintenance department included, and solved the ammunition expiry problem by keeping the production line breathing in its hands. There's one more rung on that ladder. It's an animal that moved the factory inside its own body and rents out floor space in one of its organs to a workforce that isn't even an animal. To meet it, you need to picture a tide pool in Australia and a child reaching for the prettiest thing in it. The prettiest thing in an Australian tide pool, some days, is a golf ball-sized octopus with skin the color of wet sand. And when it's disturbed, that skin does something no warning label has ever done better. 50 or 60 rings across its body and arms light up in iridescent electric blue, flashing on hard, pulsing, each ring a muscle-framed mirror that the animal flexes open and shut faster than you can blink. It's beautiful in a way that makes people reach into the water. That's the problem. That reach has killed grown men. This is the blue-ringed octopus, genus Hapalocalaena, and the weapon it carries breaks the pattern of everything before it in one crucial way. Every stolen arsenal so far had to be eaten, harvested, or held. The Blue Ring's weapon is manufactured continuously inside its own body around the clock, but not by the octopus. The toxin is tetrodotoxin, TDX, in the shorthand of the emergency rooms that occasionally have to deal with it. And it's worth understanding what it does, because it's one of the most precisely awful molecules in nature. Every nerve impulse in your body, Every thought, every heartbeat command, every twitch of your diaphragm travels as a wave of sodium ions rushing through channels in the nerve membrane. Tetrodotoxin is a plug. It's a small molecule shaped almost exactly like the mouth of that channel, and it sits in the opening the way a cork sits in a bottle. The channel can't open. The impulse dies on the wire. The consequences follow with horrible logic. Touch, then numbness. Numbness, then weakness. Then the voluntary muscles stop answering. Then the muscles that work your lungs stop answering. And a person who got an essentially painless bite from a small, annoyed octopus 20 minutes ago is now lying on the sand, fully conscious, unable to move, unable to breathe, watching. TTX doesn't cross into the brain in any meaningful way. The mind stays clear the whole time. There is no antivenom. There has never been an antivenom, and the treatment is almost medieval in its simplicity. Breathe for the victim. Ventilate them, by machine or by mouth, for as long as it takes the body to flush the molecule through, and the nerves come back online, and the person survives, usually intact. The weapon doesn't destroy anything. It switches you off and gambles that no one will keep your bellows working until it wears off. Now, where does an octopus get a molecule like that? Not from its own chemistry, as far as the evidence runs. Tetrodotoxin shows up in a bizarre scatter of unrelated animals, pufferfish, certain newts, a few crabs, some flatworms. This octopus and the common thread? There is microbial. Multiple lines of work point to symbiotic bacteria as the manufacturers, and in the blue ring, the strongest evidence sits exactly where the weapon is, cultured from the animal's own salivary glands. Look at that arrangement for a second. The posterior salivary glands of an octopus are digestive organs, spit factories, built to make enzymes that soften crab meat. The gland was already halfway to being a venom organ, as it is in many octopuses, which mostly use mild salivary toxins to subdue prey. The blue ring's conversion was to turn that organ into rented industrial space. A workforce of microbes lives in the gland, brewing one of the deadliest small molecules known, and the octopus provides housing, food, and distribution. It's the boxer crab's arrangement driven inward, a whole weapons program hosted inside an organ, with a workforce that reproduces itself, retools itself, and never runs out. The distribution end matters just as much as the manufacture. The bite itself is small, a parrot beak, often painless, sometimes leaving marks so faint that victims in the case reports didn't realize they'd been bitten until their lips went numb. The octopus uses the toxin in both directions, outward, flooding it into a bite or even into the water around a crab, which stops struggling shortly after, and inward, in a way that should change how you look at the animal entirely. The females load their eggs with tetrodotoxin. The offspring hatch pre-armed, poisonous from their first minute, defended by a chemical inheritance before they've eaten anything at all. In some species, the mother's toxin is measurably concentrated into the brood. The weapon has become a family asset, passed down like the boxer crab's anemones, except in the bloodstream. Which forces the same question the sponge-eating slugs forced, one level up. Why isn't the octopus dead? It's an animal made of nerves, one of the most neuron-dense invertebrates on Earth, soaked in a nerve poison of its own carriage. The answer, worked out at the level of the actual protein, is the same trick again: target site resistance. The octopus's sodium channels carry mutations at the exact spot where tetrodotoxin binds. The cork no longer fits its own bottles. The same class of mutation shows up in pufferfish, in the TTX-bearing newts, and in the snakes that eat those newts, evolved independently over and over, wherever this molecule became part of daily life, which tells you there's essentially one door TTX can walk through, and evolution keeps finding the same way to change the lock. And the bill? For once the direct metabolic bill looks small, the bacteria do the brewing, and that's exactly what makes this case unsettling instead of comforting. The cost has moved somewhere stranger. The blue ring's defense is a bluff. It can never test cheaply. A creature the size of a golf ball whose entire security rests on the promise that eating it is fatal, advertised by those flashing rings. It has almost no other card. It's small, it's slow for an octopus, its camouflage is good, but its body is candy soft. And everything depends on predators reading the signal and having, somewhere in their line, learned or inherited the aversion. The weapon works best precisely when it never fires, because from the octopus's side, a defense that works by being swallowed is a defense you'd rather not use. There's one more cost, and it's ours. And it explains why this animal frightens toxicologists more than sharks do. The warning system evolved for fish and crabs. It did not evolve for a species that finds blue rings pretty. The recorded human deaths, a handful mostly in Australia, mostly people who picked one up and put it on their palm or, in one notorious case, let it walk on their shoulder, happened for one reason, and aggression wasn't it. Every layer of this animal's communication assumes an audience that flinches at flashing blue. We're the one predator that leans in. So the ladder of theft runs, in full, steal the molecule, steal the loaded cell, hold the living factory in your hands, install the factory in your organs. Each rung solved the previous rung's supply problem. And all four of those animals, from slug to octopus, share one more feature that's easy to miss. They're all defenders. The stolen weapons point outward, against things with teeth. Almost none of this arsenal is used to hunt, which leaves a gap in the story. And the animal that fills it is the most heavily armed mollusk in the sea. It took the same chemical warfare strategy and pointed it forward at prey, and to deliver it, it weaponized the one body part nobody in this story has spent yet: its teeth. Every snail you have ever seen owns the same piece of equipment, a radula. It's a feeding organ, a flexible ribbon studded with rows of microscopic teeth. And the snail uses it like a rasp, pressed against a leaf or a film of algae and dragged over and over, scraping food loose. It is one of the oldest, dullest, most utilitarian structures in the animal kingdom. A tongue made of sandpaper. It has been scraping surfaces for something like 500 million years. The cone snails took that ribbon of teeth and turned each tooth into a harpoon. A harpoon in the full technical sense of the word. Pull a single tooth out of a fish-hunting cone snail and put it under a microscope, and you're looking at a hollow, translucent spear, an engineered dart with a needle tip, backward-facing barbs to hold in flesh, a folded blade that opens on impact in some species, and a channel running through its core for pumping venom. One tooth. The snail carries a quiver of them, a sac where teeth are grown, matured, and stored in sequence, moved forward one at a time like rounds through a magazine, because each harpoon, like the nematocysts the slugs were stealing, is used once and thrown away with the wound. There are several hundred species of cone snail, pushing toward 1,000 described. And they live mostly in warm, shallow water, mostly buried in sand by day, wearing shells so beautiful that collectors have died for them. That last clause is not decoration. People have picked up live cone snails for the pattern on the shell, held them in a closed hand, and been stung through the palm. The big fish-hunting species, above all Conus geographus, have killed several dozen people on record. There's a piece of grim beachcomber folklore that calls Geographus the cigarette snail, the idea being that after it stings you, you have about enough time left to smoke one. The timeline in that joke is exaggerated. The outcome often wasn't because, like the blue ring's venom, the paralysis comes for the breathing muscles. There's no anti-venom, and the only treatment is a ventilator and patience. Here's what makes the cone snail the climax of the chemical arc of this story rather than a footnote to it. The venom itself is a library of weapons, and the library was assembled with something disturbingly close to strategy. The man who opened that library is Baldomero Oliveira, a biochemist at the University of Utah who grew up in the Philippines, collecting cone shells as a boy, and who decided, decades into a distinguished career on DNA enzymes, to find out what was actually in the venom of the snails he'd been picking up since childhood. What his lab found, peptide by peptide, was that a single cone snail species carries a cocktail of dozens to hundreds of distinct small proteins, conotoxins, and that each one is a precision tool aimed at one specific piece of nervous system machinery. One peptide plugs a particular sodium channel. Another jams a particular calcium channel. Another locks the receptor that muscles use to hear commands. Individually, they're scalpels. The snail deploys them in coordinated packages, and Oliveira's group gave those packages a name that has stuck in the literature, because nothing tamer describes it: cabals. The lightning strike cabal is the first package, a set of toxins that hit the fish's nervous system like a stun gun, firing every excitable membrane at once, locking the body rigid in a fraction of a second. That buys time for the motor cabal, the slower package, which then shuts down the neuromuscular system permanently, channel by channel. Stun first, then paralyze. 2 waves, chemically staggered, out of one injection through one disposable tooth. Understand why the 2-stage design exists, because it's the same physics problem that has been haunting every hunter in this story. The snail is slow, catastrophically, comically slow. It hunts fish, animals that can accelerate away from danger in milliseconds, with a body that moves at garden snail speed. The strike itself solves part of it. The harpoon tooth sits at the tip of the proboscis, a stretchy hunting trunk the snail extends toward a resting fish with agonizing patience, and when it fires fast, driven by hydraulic pressure. The tooth punches in, and the venom goes with it in the same instant, while the tooth stays tethered to the snail like a whaler's line. A fish struck by the lightning strike cabal doesn't get its milliseconds. It goes rigid before its escape reflex finishes forming. And then the snail, unhurried, reels it in and swallows it whole through a mouth that opens like a sock. Then there's Conus geographus, which barely bothers with the harpoon first approach, and what it does instead is the single most unsettling piece of chemistry in this entire story. Geographus is a net hunter. It approaches sleeping fish at night and opens its rostrum. The whole front of the animal distends into a wide, soft funnel, and it engulfs fish that by any reasonable account should wake up and leave. They don't. And part of the reason they don't, discovered in 2015 by Helena Safavi Hemami, Oliveira, and colleagues, is that the snail releases venom components into the water around the fish before it ever touches them. Researchers have called this package the Nirvana Cabal, and its flagship component is the one that made headlines in biochemistry departments around the world. It's insulin. Actual insulin. The same hormone your pancreas releases after a meal to pull sugar out of your blood, one of the most universal metabolic signals in vertebrate life. The cone snail manufactures a weaponized fish insulin in its venom gland, an organ that has no business making hormones for export, and it floods the water with it. The insulin crosses the fish's gills, hits its bloodstream, and does exactly what insulin does: drives blood sugar down, except at overdose scale, on command, from outside. The fish crashes into hypoglycemic shock. Its brain, which runs on glucose, browns out. It becomes sluggish, disoriented, unable to organize an escape, and the funnel closes over it while its own metabolism betrays it. A hormone. The internal chemical messenger that says you've just eaten, turned into a weapon that ends with the target being eaten. Of everything in this story, the fired lungs, the thrown guts, the stolen stingers, I'd nominate this as the creepiest single conversion, because it doesn't repurpose an organ of the snail's. It repurposes the prey's own regulatory system. The weapon is a forged order, written in the victim's own chemical language, delivered through the water like a gas. And the cone snail's insulin turned out to have a property no human chemist had managed to build. Your insulin, the kind diabetics inject, is naturally sticky. The molecules clump into groups of 6, and after injection, those clumps take time to break apart before the hormone can act, which is why even the best fast-acting formulations lag behind a meal. The snail's combat insulin has no time for that. It evolved for speed of knockdown, so it lost the sticky region entirely. It's the smallest, fully functional insulin known, and it works nearly instantly. Structural biologists have since used it as a blueprint for redesigning human insulin to act without the delay. Keep that on file. It's the first deposit in an account this whole story cashes out at the end. The snail pays for its arsenal the way you'd now expect. The harpoons are single-use, the quiver is finite, and growing a new tooth is slow. A snail that has spent its ready teeth is disarmed for real, on a timescale of days. The venom is among the most metabolically expensive secretions known relative to body size, a payroll of hundreds of custom proteins that has to be met before every hunt. And the whole apparatus locks the animal into total commitment. A hunter this slow gets one strike, and a strike that misses, or a fish too big for the funnel, costs it a spent magazine and a wasted payroll with no chase possible. Chemistry bought the cone snail everything speed never gave it. And the price is that every single encounter is all in. That's where chemistry tops out. A snail that forges hormones and runs a peptide weapons library is close to the ceiling of what molecules can do for you in the sea. So the next escalation isn't a molecule at all. There are animals down there that skipped chemistry entirely and weaponized physics, and the first of them turned a claw into a gun that fires the ocean itself. Put a hydrophone in the water over almost any warm, shallow seabed on Earth, and you'll hear something that surprises everyone the first time. Frying. A continuous, sizzling crackle, like bacon in a pan the size of a lagoon, day and night, never stopping. That sound is one of the loudest and most persistent biological noises in the ocean, and it's being produced by shrimp about 2 inches long, snapping. They're called snapping shrimp or pistol shrimp, and the second name is the accurate one. Each of these animals carries 2 claws that no longer match. One is a normal little pincer that handles food. The other has been converted, swollen to nearly half the size of the shrimp's entire body, rebuilt internally, and turned into the only true gun in the animal kingdom. Here's the firing sequence, slowed down by around 200,000 times, which is what it took for researchers to actually see it. The big claw cocks open and latches, held by a mechanism that stores energy in the flexed shell and muscle, the way a crossbow stores it in the limbs, because this is the recurring trick of every fast weapon in nature. Muscle is strong, but slow, so you never fire with muscle directly. You fire with a spring that muscle spent the previous moments loading. When the latch releases, the claw's hammer half slams, shut in under a millisecond, and a plunger on the hammer rams into a matching socket on the other half, ejecting a jet of water at speeds on the order of 60 miles an hour. The jet is not the weapon. This is the part that took until the year 2000 to work out, when Michel Versluis, Detlef Loos, and colleagues put ultra-high-speed cameras on snapping claws. The jet moves so fast that the pressure inside it drops below the vapor pressure of seawater, and the water tears. It boils cold. A bubble of vapor rips into existence in the wake of the jet, a hole punched in the ocean itself, and for a fraction of a millisecond it balloons outward. Then the surrounding sea, which is still pressing in at full weight, slams the hole shut. That collapse is the gunshot. The implosion releases a shock wave that radiates outward and hits like a hammer made of pressure. The snap you hear isn't claw halves clicking together. It's a bubble dying. Measured at close range, the pulse reaches around 218 decibels in underwater terms, putting a 2-inch crustacean's handgun in the same loudness conversation as much larger and more famous ocean sounds. Small fish, shrimp, and crabs, caught within a body length or two of the muzzle, are stunned or killed outright without ever being touched. The shrimp then walks over and feeds on an animal it knocked out with collapsing water. And there's a detail on top that sounds like it was invented for a headline, except it ran in the journal Nature under the title, "Snapping Shrimp Make Flashing Bubbles." In the last instant of the collapse, the vapor inside the bubble is compressed so violently that it heats to several thousand degrees. The published estimates land in the range of 8,000 degrees Fahrenheit, which is shouting distance from the surface of the sun, and it emits a flash of light. The flash lasts billionths of a second and is far too faint to see with the naked eye. It took photon detectors to catch it. The phenomenon got named shrimpoluminescence, which is the rare case of scientists being funny on purpose. Nobody thinks the light itself does anything for the shrimp. It's a side effect, the exhaust flare of physics being pushed that hard in that small A space. A shrimp's claw, for a nanosecond at a time, contains one of the hottest points on the planet. The claw doing all this is a converted food handling pincer. That's the whole resume. The same limb that, on the shrimp's other side, is still doing its ancestral job of picking up edible crumbs, has, on this side, been rebuilt into a spring-loaded cavitation gun. And the conversion runs so deep that the animal treats the weapon as more essential than the limb. Because here's what happens when a pistol shrimp loses its gun claw to a predator or an accident. The surviving little pincer transforms. Over the following molts, the modest food-handling claw on the other side grows, reshapes, and becomes a new gun, while the regenerating limb on the wounded side comes back as the small pincer. The animal swaps hands. Its body carries a standing contingency plan for rearming itself, written into its development, evidence of how non-negotiable this weapon is to the way the animal makes a living. The sound has consequences at scales the shrimp knows nothing about. That permanent crackle is loud enough in aggregate to fog sonar, and during the Second World War, naval acousticians learned to treat shrimp beds as terrain. The crackle degraded early listening gear and gave submarines something to hide inside. Charts of snapping shrimp bottom became, briefly, a minor military asset. A defense analyst in the 20th century was studying the acoustic output of a crustacean's converted lunch-picking claw. It won't be the last time in this story that human engineers show up at the door of one of these animals with a notebook. There's even a version of this weapon that reshaped a society. In sponges on Caribbean reefs live snapping shrimp of the genus Synalpheus, and some of those species turned out to be the only known eusocial animals in the ocean. Colonies hundreds strong with a single breeding queen, discovered by Emmett Duffy in the 1990s, the marine equivalent of a beehive. And the colony's defense rests on the big clawed members holding the sponge's corridors, snapping at intruders. The gun claw made fortress life defensible, and fortress life made a queen possible. A converted pincer, scaled up in social consequence until it produced the closest thing the sea has to an ant colony. The bill is paid on a schedule, and the schedule is the molt. A shrimp's weapon is part of its skeleton, and its skeleton has to be shed and regrown every time it gets bigger. In the days around each molt, the spring, the latch, and the hammer are soft. The whole gun turns to rubber. Every pistol shrimp lives a cycle in which it periodically becomes an unarmed, soft-bodied animal whose entire lifestyle was designed around ordnance it temporarily doesn't have, and it has to survive those windows on stealth and burrow architecture alone, hiding inside its own house until its gun hardens back into being a gun. Hold on to the molt problem. Because the pistol shrimp's cousin, the animal with the most extreme version of this whole spring-loaded arsenal, has the same vulnerability. And what it does during that window is the closest thing in this story to a con job. That cousin treats cavitation as a bonus. Its main weapon is a fist. The mantis shrimp is not a shrimp, and the distinction matters more than pedantry usually does. Stomatopods are their own crustacean lineage, split off from the rest hundreds of millions of years ago, and they've spent that entire span refining one idea: a pair of front limbs folded under the face like a praying mantis's arms that started out as ordinary food-handling mouthpart limbs and became the fastest striking weapons ever measured on an animal. There are two schools among the roughly 450 species. The spearers keep the ancestral shape sharpened. The folded limb ends in a row of wicked barbed spikes, and they ambush soft prey from burrows, impaling passing fish with a strike quick enough that fishermen in some places call the animals thumb splitters. For what happens when you handle a net carelessly? The smashers went further. In a smasher like the peacock mantis shrimp of Indo-Pacific reefs, a lobster-sized creature colored like a parrot that fell into a paint factory, the tip of the limb folds back entirely, and the elbow leads. The heel of that folded limb has thickened into a calcified knob called the dactyl club. A fist, in other words. The animal punches. Sheila Patek, the biomechanist who first got cameras fast enough to resolve it, found that the club leaves its cocked position and accelerates at around 10,000 times gravity, in the neighborhood of what a bullet experiences leaving a barrel, reaching speeds around 50 miles an hour, underwater, through a medium roughly 800 times denser than air, which resists fast motion the way syrup resists a spoon. The strike is over in about 3/1000ths of a second, which is faster than the muscle that powers it can even twitch. That paradox has the same resolution as the pistol shrimp's gun, because both animals inherited the same engineering principle: never fire with muscle. The mantis shrimp's limb contains a latch and a spring. The spring is a marvel in its own right. A saddle-shaped section of the limb's shell, curved in two opposing directions like a Pringle. A geometry engineers call a hyperbolic paraboloid and use in roofs precisely because it stores and spreads stress beautifully. The animal's muscles compress that saddle slowly. The latch holds the loaded limb like a cocked hammer, and when the latch slips, the skeleton itself throws the punch. Muscle is the archer. The limb is the bow. Then the club hits, and it hits twice. This is the detail that connects it back to the pistol shrimp and elevates the whole weapon into deeper strangeness. When Patek and Roy Caldwell measured strike forces against sensors, every blow produced two force peaks less than a millisecond apart. The first is the club itself, arriving with forces measured up to around 1,500 newtons, hundreds of pounds, from an animal you could hold in one hand. The second is cavitation. The club moves so fast that water can't close in behind it against the prey's shell. A vapor bubble tears open in the gap, and its collapse delivers a second hammer blow. The pistol shrimp's entire weapon, showing up here as a free bonus packaged inside a punch. Prey gets hit by the fist, then hit again by the fist's wake. The targets tell you why this arsenal exists. Smashers eat armored things: snails, hermit crabs, clams, the walking bank vaults of the reef, the animals whose whole strategy is that opening them costs more than they're worth. The mantis shrimp made a career of disagreeing. It carries its prey to a favorite anvil rock and hammers, methodically, at the same spot on the shell, dozens of blows if needed, until the vault gives up. Aquarium keepers learned the collateral version of this the expensive way. A peacock mantis in a glass tank occasionally objects to the glass, and there are well-worn accounts of cracked aquarium walls. Public aquariums that keep them tend to do it in acrylic. That raises the question the whole weapon balances on, and it's the question that pulled materials scientists into the story. Why doesn't the club destroy itself? It's made of the same general stuff as the snail shells it demolishes, mineralized chitin, and it delivers and absorbs bullet-grade impacts tens of thousands of times between molts. The same blow that shatters a snail should shatter the fist. David Casales' group at the University of California spent years taking the club apart, and the answer has become one of the most borrowed designs in modern materials research. The club is layered like an engineered composite. The impact face is dense crystalline mineral, hard enough to deliver the blow. Beneath it, the body of the club is built from sheets of chitin fibers, each sheet's fibers running parallel, but every sheet rotated slightly relative to the one above, so the fiber direction spirals as you descend through the material. A helicoidal stack, known as a Boulligan structure. Cracks are the enemy of any hard material. A crack that runs straight through is a broken club. In a Boulligan stack, a crack can't run straight. It gets steered into a spiral, forced to twist along the rotating fiber planes, spending its energy on an ever longer corkscrew path through the material instead of a clean break. The club's whole trick is containment. It's engineered to be full of tiny, safely imprisoned cracks, and at the next molt, the whole scarred weapon is thrown away and grown fresh. There's the molt again. And for a mantis shrimp, the molt is a catastrophe of a very specific kind, because this is the most heavily armed animal on the reef. It lives in permanent real estate disputes over burrow cavities, and for a window of days, it can't throw a punch at all. Its spring is soft, its club is soft, its skin is soft. Every rival on the block would love the cavity it's holding, and its actual fighting capacity has just dropped to approximately 0. What it does during that window was documented by Ron Caldwell and Steven Steger in a study that has become a classic in the science of animal communication, because it's one of the cleanest demonstrations of dishonesty in nature. Mantis shrimp threaten each other with a display called the Merrill spread, the raptorial arms swung wide, presenting a colored badge on the inner face of the limb, the stomatopod equivalent of showing your holster. It's normally an honest signal backed by a working weapon. And newly molted mantis shrimp, the ones who physically cannot strike, do not go quiet. They display more. Challenged in their burrows, at their most helpless, they throw the Merrill spread harder and more often than armed animals do, advertising a weapon that is, at that moment, a soft sculpture of a weapon. And it works. Intruders, who can't verify the club without risking a hit from what might be an honest signaler, mostly back off. The bluff succeeds precisely because the badge is usually true. Notice what that means. The weapon has become detached from its own use. The club's reputation now does work. The club temporarily can't. The animal is spending the accumulated credibility of every real punch ever thrown by every hard-shelled rival its species has produced. That's a weapon operating as pure information. No contact, no venom, no physics. A threat floated on borrowed collateral by a creature in a bathrobe. And once a weapon can work as information, the door opens to weapons that are nothing but energy and information. No club, no jet, no barb, nothing mechanical crossing the gap at all. So this story arrives at a fish lying under the sand with its eyes on the top of its head, and to the strangest fate any muscle in this story has suffered. The muscles this next animal weaponized are the ones that were built to move its eyes. The fish is called a stargazer, which is one of the gentlest names ever wasted on an animal. It's named for its anatomy. The eyes sit on the very top of a flattened, armored head, staring permanently upward at the sky. The reason they point that way is that the stargazer spends its working life buried. It swims down to a sandy bottom, shivers its body, and disappears in seconds, sand flowing over its back until nothing remains at the surface but two pinhead eyes and the rim of an enormous upturned mouth, a mouth that opens toward the sky like a trap door set in the seafloor. Then it waits. Some species sweeten the wait. A thin filament grows from the floor of the mouth, and the buried fish flicks it above the sand, where it wriggles like a worm. A small fish comes down to take the worm. The seafloor opens. The stargazer's mouth inflates upward in a few milliseconds, and the water above it, along with everything swimming in that water, is simply relocated inward, buried again before the sand settles. That alone would earn it a place in an ambush documentary, but not in this one. What earns it a place here is a pair of structures sitting in pouches just behind those upward eyes. Naturalists handling stargazers have been startled for centuries by the discovery, made the hard way, that some of these fish shock you. Grab an American stargazer of the genus Astroscopus bare-handed, and you can take a jolt. The fish generates genuine electrical discharges, reported at up to around 50 volts. From organs it can fire at will. And when anatomists traced where those organs came from, the answer was almost indecent: their eye muscles. The electric organ of Astroscopus develops from extraocular muscle, the small, fast, tireless muscles that in every other vertebrate, including you, do nothing but point the eyeball. In the stargazer's lineage, a set of that tissue stopped being muscle and became a battery. The conversion is worth walking through because electric organs are perhaps the purest illustration in all of biology of what this entire story has been about. A muscle cell and a battery cell turn out to be separated by a shockingly short evolutionary distance because a muscle cell already is an electrical device. Every contraction begins with a voltage pulse across the cell membrane. The mechanical part, the shortening, is downstream machinery. Evolution's move, made independently at least half a dozen times in fishes, is brutally simple. Throw the downstream machinery away. The cells flatten into discs, abandon their contractile filaments, keep the electrical excitability, and stack face to face in columns, like coins in a wrapper, like the cells of a battery in series. Each disc contributes about 1/10 of a volt. Stack hundreds, fire them in perfect synchrony with a single nerve command, and the voltages add. The tissue can no longer move anything ever again. What it does instead is discharge. That's the price paid in the clearest currency yet. Every other conversion in this story kept some shadow of the original function. The sea star's stomach still digests, the hagfish's skin still coats. An electrocyte is a muscle cell that gave up the entire concept of movement. The organ sits there, structurally muscle, biochemically muscle, doing nothing a muscle does. In the stargazer, tissue that was built to aim an eye now defends the animal instead. A modest, snappy jolt, nothing lethal, but startling enough to convince whatever just bit down on a buried fish that it has made a category error. Researchers still debate how much the discharge helps in actual hunting versus pure defense. The honest reading of the evidence is defense first, and the stargazer stacks its deterrence. Many species also carry a venomous spine above each pectoral fin, making this one of the very few animals on Earth that is simultaneously venomous and electric, a buried trap with 2 different weapon systems and eyes on the roof. There's a reason writers have called the stargazer the meanest thing in creation. It's unfair, but you can see how they got there. 50 volts, though, is the hobbyist version. The industrial version swims on the same sandy bottoms, and humans have known about it for as long as there have been fishermen in the Mediterranean. The torpedo ray is a soft, rounded, sluggish ray. No spine on the tail, no speed, no apparent defense at all. And on each side of its head, filling most of its disc, sits a kidney-shaped electric organ built from converted gill arch muscle. Same trick, different donor tissue, scaled up. Stacked columns of electrocytes numbering in the hundreds of thousands, wired in parallel to push real current through seawater. The big Atlantic torpedo has been measured delivering discharges around 200 volts, enough to knock a diver's regulator loose, enough to stun outright the fish it hunts. And that's the detail that separates the torpedo from the stargazer. The torpedo hunts with it. It drifts over the bottom at night, wraps its disc around a passing fish like a blanket, and fires. The prey convulses and goes limp inside the embrace. A slow, unarmored, toothless animal eats fast fish because the weapon crosses the gap at the speed of an electrical field, and no reflex in the sea outdraws that. The Romans knew, and what they did about it belongs in this story's ledger. The physician Scribonius Largus, writing in the 1st century, recorded a standing prescription for chronic headache and for gout. Take a live black torpedo, place it on the patient, on the head for the one, under the feet for the other, and hold it there through the discharges until the pain numbs. It worked, at least sometimes. For the same reason modern electrical nerve stimulation therapy works. Strong stimulation scrambles and dampens pain signaling, which means the first electrotherapy device in medical history was a converted fish, applied whole, 2,000 years before anyone knew what electricity was. The name runs the other direction too. Torpedo comes from the Latin for numbness. The ray was named for the effect, and when naval engineers much later built a weapon that struck invisibly from underwater, they borrowed the fish's name for it. The fish had the name first. The weapon borrowed it. An electric organ is close to the end of one road. A weapon with no moving parts, no chemistry crossing the gap, no contact, just energy shaped by an organ that gave up its old life completely. There's one conversion left in this story, and it's the mirror image of invisibility. It's a weapon made of pigment. Its whole job is to be seen and to lie. Squid ink is one of those things familiarity has ruined. It's on restaurant menus, it colors pasta, it's a garnish, and because we mostly meet it as food coloring, almost nobody stops on the fact that the substance itself is one of the strangest weapons in the sea, a weapon whose active ingredient is the same molecule family that colors your own hair. The pigment is melanin, real melanin, built by the same enzyme, tyrosinase, that your skin uses to tan. In you, the job of that pigment is sunscreen and coloration. A squid builds it by the gram, in a dedicated gland folded against the end of its digestive tract, and stores it in a sac. And the position of that sac tells you its history, because the ink exits through the same plumbing as everything else, leaving the animal. Another rear-facing weapon. The ocean keeps arriving at that answer, and at this point in the story, you can see why. The back end is where the pursuing mouth is. The lazy image of inking is the smoke screen, a black cloud, an escape behind a curtain. And squid and octopuses do use it that way. The version worth this chapter is more precise. Watch a squid under real attack, and you'll often see it do something with an almost theatrical structure. The animal, dark-colored, jets away from the predator, then blanches, dumping its skin pigments to near white in a fraction of a second, and at the same moment releases a package of ink bound up with mucus. That package doesn't billow, it hangs. It's called a pseudomorph, false body, and it's a blob roughly the size and shape of the squid that made it, holding together in the water like a ghost, dark and squid-shaped, exactly where the squid just was, while the actual animal, now pale as glass, is already yards away and accelerating. The predator's eye does what eyes do. It stays on the dark thing. Fish and birds have been documented attacking pseudomorphs outright, committing to the phantom, mouth first, and by the time the ghost disperses across their face, the real squid is gone. That's pigment doing the work of a body double. The molecule that says, "Here I am," released as a lie about where I am. There's chemistry folded into it too. Cephalopod ink is not just pigment. It carries a payload of free amino acids and pigment precursors, including dopamine and its relative L-dopa, molecules that are, in other contexts, neurotransmitters. What all of that does to a predator is still being worked out. What's documented is that ink acts on chemical senses, not just eyes. It can jam or repel the smell and taste apparatus of hunters like moray eels, and squid themselves treat drifting ink from another squid as an alarm signal and take evasive action. One substance, running visual deception, chemical interference, and public warning at the same time. And you can watch evolution price this weapon by where it cancels the program. In the deep sea, below the reach of sunlight, black ink is a product with no market. A black decoy in a black world is nothing. The deep-sea octopus lineages have, accordingly, lost the ink sac entirely. The organ was dropped from the budget. One small deep-water squid, Heteroteuthis, rebuilt the concept for the dark instead. Its escape secretion is luminous. Startled, it ejects a cloud of glowing fluid, a flare, a burning ghost hanging in the blackness for the predator to investigate, while the animal itself vanishes into the dark it was built for. Same tactic, inverted physics. Where light is absent, the decoy brings its own. The strangest pigment weapon of all, though, belongs to an animal most people have never heard of, and it closes this arc by combining nearly everything this story has established: the theft, the diet lock-in, the chemical forgery, into one soft purple package. The sea hare is a sea slug the size of a rabbit, and roughly that shape, with two ear-like sensory tentacles that earned it the name. Aplysia, to scientists who know it intimately because its enormous nerve cells made it the workhorse of memory research. It's slow, even by slug standards, essentially defenseless to look at, and it grazes red algae in the shallows. Provoke one and it releases a shocking quantity of vivid purple ink, and that pigment is stolen. The purple is manufactured from phycoerythrin, the light-harvesting pigment of the red algae it eats, the molecule that once caught sunlight for a seaweed repackaged as ammunition. Feed a sea hare a diet without red algae, and its ink runs thin and pale. The weapon is diet-dependent, exactly like the nudibranch's stolen stingers, a magazine that empties unless the animal keeps eating the right prey. What the ink does is where it gets properly devious, and this was worked out in careful experiments by Charles Darby's group at Georgia State University using sea hares and spiny lobsters, one of their standard predators. The purple ink comes out alongside a second secretion, a viscous white substance called opaline from a separate gland, and the mixture attacks the lobster on three fronts at once. The cloud screens, like any ink. The opaline physically gums the lobster's antennules, the smell and taste organs, coating the very sensors it would use to relocate the prey. And the third front is the remarkable one. The mixture is loaded with free amino acids at concentrations that read, to a lobster's chemical senses, as food. The lobster's feeding program fires. It stops hunting, seizes at the cloud, grooms, mouths the water, behaves for all the world as though it's already eating because chemically, it's being told that it is. Researchers named the tactic phagomimicry, the fake meal. The sea hare escapes by serving the dinner early, a chemical forgery of the reward, cached by the predator's own reflexes, while the actual meal crawls away. Add it up and the sea hare is running the cone snail's trick in reverse. The cone snail forges the victim's internal signals to shut it down. The sea hare forges the attacker's victory signal to switch it off. Both weapons are lies written in molecules, and both work because a nervous system, anywhere in the ocean, ultimately believes it's chemistry. That's the last conversion, and it lands the whole arsenal somewhere worth standing back from. This story started with hardware, a lung fired at a crab, and it ends with weapons made of nothing but information. A pigment that impersonates a body, an amino acid broth that impersonates success. The raw materials never stopped being ordinary: skin, ligament, spit, teeth, muscle, ink. What escalated animal by animal was what counts as ammunition: matter, then machinery, then energy, then information. So why does the ocean's armory look like this? Why is the inventory a fired lung, a weaponized boundary layer, a ligament that obeys orders, a stomach used outdoors, a stolen poison, a stolen gun, a hostage with tentacles, a rented bacterial weapons plant, a tooth become a harpoon, a hormone become a forgery, a pincer become a firearm, a mouth part become a fist, an eye muscle become a battery, and a pigment become a lie? Because evolution has no clean sheet of paper. That's the machine under everything you've just seen. Natural selection can't invent an organ from nothing. Mutation can only edit blueprints that already exist, which means every new weapon in the history of life began as something else. Some organ with a full-time job, drafted sideways into violence, because a modified copy of it happened to save one animal's life one time, and that animal bred. Biologists have a word for a trait caught working a job it wasn't originally shaped for: exaptation. The whole arsenal in this story is exaptation with teeth. Nobody designed a slime gland for shark defense or an eye muscle for delivering shocks. Ordinary equipment got cornered, and the cornered version that survived was the one whose spare capacity turned out, under pressure, to be a weapon. And that's also why every single one of these weapons came with a bill attached, without exception, anywhere in the story. A designed weapon can be optimized in peace. A converted one is always double booked. The organ owes rent to its old job while paying for its new one. The sea cucumber breathes worse for weeks because its defense was made of lung. The hagfish runs a month on an empty magazine because its ammunition is made of cells that die on delivery. The nudibranch and the sea hare are chained to specific meals because their weapons are made of groceries. The boxer crab eats clumsily forever because its hands are holsters now. The stargazer carries muscle that will never move again. The mantis shrimp is periodically reduced to fraud because its entire arsenal is part of a skeleton that has to be thrown away on schedule. Conversion is never free. The bill is the signature of the process, proof that the weapon was remodeled out of something the body still needs. Here's the part that should change how you look at the water, though. None of this is finished. Every system in this story is one frame of a race that is still being run, right now, at full speed. The sponges are still in the fight. They remain under continuous selection to shuffle their chemistry into something the slugs can't sort, store, or survive. And every sponge generation that gets eaten is pressure toward a new molecule. The slug's sorting machinery chases the chemistry in turn. The hydroids keep building nematocysts. And every capsule that fires into a slug's mucus instead of a slug's mouth is selection working on both sides of that membrane at once. Nobody yet fully understands the slug's immunity, and it's entirely possible the immunity is itself a moving target, because the stinger it has to beat next century won't be the stinger it beats today. Tetrodotoxin is a standing arms race in molecular form, a lock, a key, and a long record, written independently into pufferfish, octopus, and everything that eats them, of channels mutating away from the poison and the poison's carriers ratcheting the dose. Even the mantis shrimp's bluff keeps shifting under it, because bluffing only works while it stays rare enough that the badge is usually honest. Push the fraud rate up, and the display collapses into noise. So the honesty of the signal is under permanent negotiation, every burrow dispute a tiny vote on whether the currency holds. Nothing in the sea has won. Nothing gets to win. A defense that becomes perfect starves its predators into finding new prey or new tricks. and the pressure resumes from a new direction. What you've been watching for this entire story is a live negotiation, hundreds of millions of years deep, in which every creepy converted body part is simply the current draft of an argument that neither side is allowed to leave. Which raises a question I want to put to you directly, because I've gone back and forth on it the whole time I was putting this together. Out of everything in this arsenal, the commanded ligaments, the stolen stingers, the rented bacteria, the forged insulin, the cavitation gun, the eye muscle battery, the fake meal, which conversion is the one that actually unsettles you? The one that got under your skin? Put it in the comments and say why that one landed for you, because the answers to that question split people more than you'd expect. And they're half the reason I ask. If this kind of deep dive is your thing, the like and the subscribe are how you tell me to keep making them. There's a lot more ocean. And here's where this actually lands, because there's a final escalation in this arms race, and it's happening above the waterline. A third party has entered it, us, as students this time, and increasingly as clients, because it turns out that when an unsupervised research program runs for 500 million years with extinction as the penalty for bad designs, the survivors are carrying answers to problems human engineers are still stuck on. And over the last few decades, quietly, lab by lab, we've started collecting. Start with the cone snail, because that account has already paid out. One of the calcium channel peptides from the venom of Conus magus, a fish-hunting cone from the Pacific, was developed into a drug called ziconotide, approved for human use in 2004 under the name Prialt. It's a painkiller for the worst cases, the cancer and nerve damage pain that morphine stops touching, and it works by doing in a human spinal cord a controlled version of what it evolved to do in a fish, blocking specific channels that pain signals need to pass. It's enormously potent by weight compared with morphine, and because it isn't an opioid, it doesn't build tolerance the way opioids do, and doesn't recruit the addiction machinery at all. It has to be delivered by a pump directly into spinal fluid, which tells you how far from a casual medicine it is, but the fact stands. And it's worth saying plainly, there are people alive right now whose pain is being managed day by day by a weapon, a snail, aimed at a fish. And that was one peptide. The cone snails as a group carry a combined library estimated in the hundreds of thousands of distinct venom components, almost entirely unscreened. Each one a pre-optimized key for some lock in a nervous system, pain, epilepsy, nerve injury. The pipelines are working through it now. Meanwhile, the geographus insulin, the forged surrender order, is doing exactly what was promised earlier in this story. Its stripped-down architecture, the fastest-acting insulin ever found, has become a structural blueprint in the effort to build faster insulins for diabetics, an arms race molecule teaching us how to shorten the minutes between injection and effect that diabetics have been managing around for a century. Even tetrodotoxin, the Blue Ring's rented poison, has spent years in human clinical trials in carefully microdosed form as a non-opioid painkiller. The logic being that a molecule this good at silencing sodium channels might, at 1,000th of the terrible dose, silence only the ones screaming. The sponges have already put drugs on pharmacy shelves. The chemistry that arms the nudibranchs, that same shelf of defensive molecules, yielded the compounds that led to cytarabine, a backbone of leukemia treatment for decades. Developed from unusual nucleosides first found in a Caribbean sponge, it yielded halocondrin B from a Pacific sponge whose synthetic descendant eribulin treats advanced breast cancer today. Latrunculin, the slug's stolen weapon, sits in catalogs as a standard cell biology tool. The war chemistry of animals that can't run turned out to be, molecule for molecule, the best drug discovery library on Earth, and it was assembled by predators voting with their appetites. The materials people are at the door too. The mantis shrimp's dactyl club, the spiral-stacked, crack-imprisoning Bouligand architecture, has been copied into carbon fiber composites in aerospace and impact protection labs, panels that survive strikes, that shatter conventional layups, with obvious futures in body armor, helmets, aircraft skins. The hagfish's thread cells have had defense money behind them for years. Those protein fibers rival spider silk in performance. A single animal carries miles of them, and groups including the U.S. Navy's own researchers have synthesized versions of the slime itself, an instantly expanding, water-stopping net with applications you can imagine without help. The sea cucumber's mutable collagen, the tissue with a stiffness dial, directly inspired a family of synthetic composites that switch between rigid and soft on cue. One of the first was built as a brain implant electrode, stiff enough to insert, then softening in the wet warmth of living tissue to sit gently among neurons. Exactly the trick the dermis of a sea cucumber has been performing since before there were brains to implant into. The Cuvierian tubule's instant wet adhesion, the glue with no glue, is a live research target for surgical adhesives because an operating room is precisely the warm, wet, unforgiving environment where our best tapes and epoxies fail, and a sea cucumber's defense does not. There are groups studying the nematicist's eversion mechanics for microneedle drug delivery, and engineers building soft, stacked, electrocyte-style power sources, batteries organized like an electric organ, salty gel cells in series, meant to one day run implants inside the body's own wet chemistry where conventional batteries don't belong. Notice what every one of those projects has in common. Nobody is inventing, they're reading. The hard part, the search through the space of possible molecules, mechanisms, and microstructures, was already run, at a scale no laboratory can approach, financed in the only currency evolution accepts. Every compound was tested against billions of subjects. Every structure was load-tested for millions of years. Every failure was deleted. What's left in the water is the printout of the largest weapons development program in the history of the planet. And we have sequenced, imaged, and understood only the thinnest first pages of it. So that's the real ending. And it isn't an ending at all. Tonight, on some dark reef, a sponge is being eaten by a slug that shouldn't be able to eat it. And somewhere in that sponge's descendants, a molecule is already changing. A hagfish is rewinding a foot of thread inside a single cell. A cone snail is growing a new tooth in its quiver. And in a lab 6 time zones away, a peptide from its cousin's venom is being weighed for a clinical trial. The race that built every weapon in this story is still running in both places at once. In the water, where it never stopped. And now on the bench, where we've finally learned enough to hold the stopwatch. The creepiest body parts in the ocean are the receipts of that race. And the next drug, the next fiber, the next impossible material is down there right now, holstered in something soft and slow and easy to swim past, waiting for someone to look closely enough to steal it. It would be the oldest move in the book. Everything down there steals its best weapons. We're just the newest animal to figure that out.